Welcome to video 3 of the Search for Better Health series. In this video, we're going to be looking at pathogens in food and water, which formed part of your secondary source investigation, and it will also form some of the information you will need for your first-hand investigation. When we're looking at water quality, we look at water quality in terms of these characteristics. So we look at turbidity, dissolved salts, conductivity, pH, hardness, temperature, dissolved oxygen, color, taste, and odor. And then we look at the chemical characteristics, so whether there's any free chlorine, any inorganic chemicals, which are usually dissolved salts, or any organic compounds. And then we're also looking at the microbial characteristics. So we're looking for things like pathogens and cyanobacteria. To ensure that our drinking water remains safe, there are a few methods that water treatment facilities undertake to ensure that the water that comes out of our taps is clean and safe for us to drink. So one of the first few steps is filtration, and usually it starts with filtering out any big particles. So because our water, especially in Sydney, is coming from dams, so we're filtering out any leaves, twigs, bark, any of those kind of materials. But then we can also go to finer filtration where we're using mesh screens. There is some coagulation where we add in some chemicals that allow materials to join together and create flocks. Then through this flocculation that has been created is removed in one big chunk rather than little bits and pieces which might fall through the screens. And then there is again a finer filtration using sand and rock particles. One of the second processes is a disinfection, and this can occur using a few different processes and a few different chemical compounds. So we have chlorination, we have chloramination, we have ozone, and we have UV light, just to name a few. So the way in which chlorination works, which is the most common process of disinfection, is that the chlorine that is put into the water breaks down. These chemicals that it breaks into affects the lipids that are in the cell wall of bacteria, which can affect the enzymes that the bacteria produces, which renders them harmless. So they're no longer able to move about and infect people. Therefore, we're disrupting their life cycle and we're preventing any future transmission or any diseases resulting from those pathogens. And then we've got fluoridation, which doesn't actually remove any pathogens, but it's under the advice of New South Wales Health. So in New South Wales, we add fluoride, which is known to prevent dental decay. These three processes occur similar to a flowchart as is depicted here. And the reservoir which we have here, so Sydney Water purchases their water from the Sydney Catchment Authority, and that's our reservoir. So we have a few dams, two of which are the Warragamba Dam and then the Illawarra Dam. So they're our most local reservoirs. And then we can see as we go through the processes which were just discussed do occur. These processes are all in place in our water treatment facilities because of the Australian Drinking Water Guidelines. And these are some standards that regulate the water in which we drink and the water supply industry. And it ensures that we are always provided with good quality drinking water. The good quality, which we're talking about, means that these guidelines always are on the side of safety, so that they always make sure that the low levels that they're looking for of certain chemicals or pathogens in water are much lower than what's considered safe. As long as those levels in our water that is being sent to homes is below that, then they're considered safe. And these guidelines are both health related and aesthetic. So for health, we're making sure that no one can actually get sick from it. And then aesthetic is to make sure that the water that is being sent for distribution looks, smells, and tastes nice, I guess. When we talk about microbiology, which is, the study of microorganisms and how they affect and used in society, which we recognize the fact that they are present in air, water, and food, and that they have the potential to cause human disease. They also may be beneficial, and then some microbes have no effect at all on humans. When microbes reproduce, they can form colonies, and these become visible to the unaided eye, which is why we see mold on bread. These colonies can be distinguished by a few different factors, and because of these factors, we're able to distinguish whether some colonies are bacterial colonies or fungal colonies. And this is what we're looking for when we do our first-hand investigation. So here's a quick diagram which we're going to look at when we do our investigation when we grow some microbes. This is what bacteria may look like. So we can see that they can form different types and shapes of colonies. We can see that these colonies, their margins may look different. We can also see that they might be different elevations. And then a few other things have got to do with colour and whether they are transparent or opaque and those kind of things. So this all forms some valuable knowledge for our first-hand investigation, which we will complete in class. Make sure you tune in for video 4.